Hi, 7th graders from Ames Middle School. We got lucky today, and instead of just getting to chat with Ruth, who's standing over there casually watching, we're going to get to chat with some of the people in the lab who actually help make the GMOs here in Ames.
I just said these guys do GMO work here in Ames. Well, that's pretty close to accurate because they do select for specific traits and try to get them in soybeans. They do it through breeding, so it would be more accurate to call them breeders than genetic modifiers.
Could you introduce yourselves?
My name is Danny Singh. I'm glad you're watching this video. We love science. I hope some of you will take science as a career. I'm a plant scientist working at Iowa State University.
Sweet.
Hi guys, my name is Jonathan Shook. I'm a student with Dr. Singh here, and I'm working on trying to identify exactly what's causing the differences in how soybeans perform. So looking at the molecular causes of that and finding out what different genes are doing within the crop.
Sweet. So you figure out what genes do what.
Exactly.
Awesome.
Hi, my name is Kevin Chiteri and I'm a student with Singh. And in addition to what John said is that so we just help empower the plants so that they can survive when it's too hot, when it's too cold, when they are affected by insects.
Yeah.
Yeah.
So you help the traits be better for their environment.
We're
Help
trying
the
to make
traits survive.
soybean be more productive.
Yeah, so the farmers can make more
So,
money
yes.
and we can get more food.
They're environmentally conscious.
Sweet. So we're in a lab right now. There's a whole bunch of stuff going on. And the lovely people we were just introduced to are going to kind of show us around a little bit. So let's take a look.
So generally speaking, the lab is a very active place. A lot of people are in the lab working on different projects.
Group work is a thing, guys.
Group work is a thing. We work as a team. There are lots of different people in the lab. So as an example, this would be what would be a workstation. So a person, a scientist would be here. They would be doing their research, conducting their research, working with different aspects of the plant cells. So they may be looking at DNA, they may be looking at RNA, they may be looking at protein. So a lot of equipment and instruments, and I'll have John and Kevin explain a little bit more about some of the things that are going on in the lab.
Sweet.
Turns out we don't have to go all the way down there. So this is what's called a gel electrophoresis setup.
Whoa, that's a big word.
Right. So this is the PCR. So you're putting DNA into a gel, and then putting electrical current through it so that it moves throughout there. Small pieces are going to move more because they're more strongly pulled by that electric current, while the big pieces stay back further.
So it's kind of like a race.
Exactly.
And they're running a race, and the little ones have less to move, so they win that race.
Yep.
And the electricity, so we have some cords hooked up to this. The electricity is helping them run that race and push the little ones, right?
Yep. So what you're going to get in the end is you're going to have distinct bands that you can see where you can visualize that DNA, and you get bands running across for each lane that you're running. So you can run maybe 30 at a time and be able to see which combinations of alleles that are within each one of those lines. So you can identify if something's a hybrid or if it's purebred or inbred.
Yeah, okay. So those are more things we learned about in class. So what else is there?
Alright, when John also said that, so you visualize using the machine, right? But this right here
So this thing, what's it do?
is the machine that you're able to visualize the gel.
So
Once
this
it's
helps
the
us
gel-
see stuff.
Yeah, you can see
Okay.
because you can't see it just with your bare eyes.
Okay.
So you get in this machine and then you can be able to visualize the bands.
So do we see that twisted ladder shape?
No, you won't see this, right? Your teacher just told me you won't see this. You just see like bands.
Okay, why can't we see it?
They're too tiny for
Too
our
tiny
eyes.
for our eyes.
Right.
Yeah, super duper small.
It's pretty small.
Yeah. Okay, sweet.
Yeah. So what else we wanted to show you, because this is a molecular lab and this is the molecule. So it's called the molecular lab as basic things that you must have. And one of them is the fridge, which is at plus 4 degrees Celsius.
So that's chilly.
So that's 40 Fahrenheit.
Yeah,
So
40
kind of
Fahrenheit.
like your fridge at home.
Right.
Kind of like your fridge at home.
Right. And then
So what's this one?
and then we have a freezer here. You thought Iowa is cold, but this is like, can you see that? Negative 79 degrees Celsius. That
A
would be
little
like-
bit.
Negative 112 Fahrenheit.
Exactly.
Wow, negative
Right.
112.
Right.
And we think today is cold.
Wow. Right.
Yeah.
So we do store here our samples because they are sensitive to temperature changes, and so once here we know they're well stored.
I wonder what kind of ice cream you could make in a negative 80 degree freezer.
I don't know. Maybe
Yeah.
you guys make and bring us right here you see is the cleaning station.
Mhm.
So just like at home, we have a place where I'm sure you guys know what this is, right?
Maybe.
Yeah.
We don't get to do very many labs.
Okay, but that's
Seventh
okay. So
grade.
you have a station where you clean your stuff all the time.
Mhm.
Yeah, and many more instruments that we have in the lab, and we'll be happy to host you guys to come and show you more.
Sweet. So yeah, thank you guys so much. Let's say goodbye to our 7th graders.
All right.
Bye.
Bye.
Hi.
So, this is Ruth McDonald.
Hi.
And I've been told you know a lot about GMOs.
I know a little bit. Yep.
Oh, okay, cool, sweet. Because we have some questions for you today. First of all, can we ask a little bit of a question beforehand of what exactly your role here is at Iowa State? What exactly your job is? What
Sure.
exactly you do?
Sure. I'm a professor here at Iowa State University, and I am in a department called Food Science and Human Nutrition. And so our work in that department is to understand how food helps your body. And we also understand what food is, and we make different kinds of food. You might see behind me on the shelf there is some popcorn. We have one of our students that graduated, and she works at ConAgra, and she made that popcorn. She created that variety of the microwave popcorn called Boom Chicka Pop.
Sweet.
And so that's what some food scientist people do as well.
Okay, so you would know pretty much anything about if the food is safe, how it might affect you, and where it came from, right?
Correct.
Sweet. Okay, cool. I know there's a lot of questions in here. These.
Okay.
So, how many GMOs are there?
Good question.
How many GMOs are there?
So if you're talking about food that
Mhm.
has a GMO trait in it, there's
Okay.
only a few.
Only a few. Wow, I
Right.
thought there'd be a lot.
You would think there'd be
Wow,
a lot, but
things
there
I
are
was
not.
wrong with.
So
Yeah.
you can count them on your hand. There's corn, soybeans, sugar beets, alfalfa, which we don't eat but we feed to cows. There's a squash. There is an apple. There is a salmon. There is I'm getting down to my list here. I think that's mainly it right now that are in the U.S.
But I know a lot of food has changed over time. So what's the difference between a GMO one and then all the other different ones we saw?
Right. So a lot of the foods that you see in the grocery store, if you go to the produce aisle, you see lots of different colors of
Mhm.
apples, you see different sizes of oranges. All of those were genetically modified by breeding.
Okay.
So not using molecular biology tools, but just selecting and getting the traits that you
So
want.
could we still kind of call that a GMO, or they just don't?
Technically, it's not.
Technically, it's not.
just a
Okay.
conventionally bred
Okay.
food.
Okay.
So the ones that are listed as GMO are the ones that I just described.
Okay. Sweet.
Not very many.
Not very many.
Oh, and papaya.
didn't know that.
Forgot papaya.
Yeah, the ring spot.
Right.
Yeah. So that's something I am learning just
Mhm.
now. Okay, sweet. Do you like McDonald's?
Okay, this is a trick question.
Oh.
So my name is spelled MacDonald, not MC.
But do you like
Sure,
McDonald's?
McDonald's is great.
Okay. Are GMOs bad?
So this is a question that you get a lot, right? Are GMOs bad? Should I not eat them? Should I be scared of them? And the answer is no. There's never been any evidence that the foods that we have eaten in the U.S. that have been genetically modified since 1996,
And you can count them on your hands.
not very many, have any cause of causing anybody any bad health or allergic reactions. There's no evidence of that. So the government has said they're safe. Most science organizations have said they're safe. And so there isn't any reason to think that they should be harmful.
Cool.
Let's take this one with the lovely tree on
Oh,
it.
okay. Sweet. So there are two here. Let's choose one of them. What made you interested in GMOs?
So I found out that there were a lot of people that were scared about GMOs. And I started hearing consumers and people that are buying food saying, we don't want GMOs, we think they're bad. And I knew that that wasn't true because as a scientist, I knew the science and I knew what they were and how they were made. And I wanted people to understand that. So I started giving talks and reaching out to people about what the science said about GMOs so that they would feel more comfortable about understanding what's in the food and not being afraid of the science.
So you wanted people to stop being afraid of safe food.
Right. I don't think anyone should be afraid of our food because it's very safe.
What is your favorite color?
Tricky question. I don't know. That's a tough one. I think I like orange.
Orange? Orange is a good color. Alright, there's a few on this one. Let's choose this. How much does it cost to create a GM plant?
Yeah, that's a good question.
Yeah.
It costs a lot.
A whole lot.
It costs a lot. You have to think about the very start of the process. We have to identify a gene. You have to figure out what it does, where it is in the plant or the
So you gotta
bacteria.
pay all the scientists.
Figure all the science out about that. Then you have to figure out a technique to actually get it into what you want to get it into. You have to show that it got in there. You have to prove how much is in there. And then you have to do all that science stuff in the lab, and then you have to take it out into the field. And you have to plant the crop, whether it's corn or soybeans. You have to plant it, see if it grows. Does it work okay? Does it make enough corn and soybeans to make it a commercially viable product?
So I have to pay to plant it.
Plant it. And that takes years, right? Because
Yeah.
you have to grow the plant, you have to test it, then you have to grow another plant and make sure it's
safe.
consistent.
Too.
And then you have to test the safety, right?
Yeah.
You have to do a lot of experiments to show that it's still in the plant and where it goes in the environment. You have to show that it doesn't get lost, that it doesn't change.
So test the safety.
To safety, and then you have to show that it doesn't affect animal health. So you have to do some experiments with rats and mice to make sure that they're healthy when they eat it. And then you have to figure out how the government's going to regulate it. So there's rules and
figure out the
laws
rules
about
and regulations. Wow.
getting approval for that. So it takes probably 10 years.
Probably 10 years, and we have to pay a whole bunch of people 10
All
years
that
of
time.
salaries. Would you say it's in the millions?
Millions
In
of
the
dollars sometimes. Yeah.
For making one GMO plant.
Right.
Yeah.
That's why there's not a lot of them.
Yeah, how many are there?
Well, there are lots
A
of
lot
them
of
that have been developed, but the commercialization
Okay.
of them is very limited.
Like-
Probably 10, 12
Okay.
that are commercially
So not
in
many.
the market. Right.
Okay. So, is your job fun?
Yes,
Yes. Yes?
absolutely.
Okay. So, what is a GMO?
Right.
So, what exactly is a GMO?
Right. So a genetically modified organism, and what we're talking about is that in a food. So we've taken a food item. Let's just use an apple as an example. So we've taken an apple and we've changed the seeds of that apple to express a gene that in this, let's say we make it be purple. Make
Okay.
purple apple.
So, color.
That doesn't
Simple
exist.
gene.
It's just hypothetical. But you could do that.
Let's say there was a flower that can be purple. So
Right.
it has that
You took
instruction
that color
to be
in there
purple.
to make it purple.
Mhm.
And then we actually can do this with potatoes. You've seen purple potatoes. But let's just say we're going to do it in the apple. So
Okay.
we've taken the gene and we put it into the apple so that now we have a purple apple.
Okay.
That would be genetically modified because we've inserted a gene that wasn't there before and it's creating something new.
So you're the apple. I'm taking the gene from the potato or the flower that's purple, and I'm giving it to you.
Correct. And now
Yes.
I'm a new thing.
Yes.
Now I'm a purple apple. So
New
I'm a
DNA.
GMO. And it's been done using the tools of genetic manipulation.
All right. Oh, this is a little teeny tiny one. So what's it like working with them, meaning GMOs and food safety? What's it like working with GMOs?
So with any science, it's fun, right? Because you can do these tools that we have in molecular biology are amazing. I think we talked about insulin. Remember, that's the drug that people take who have diabetes.
So at the start of the video, it said insulin was something super widely used and super widely well thought of. We like our insulin drug. We like people not to be sick.
Right, and that's a genetically modified drug. So we make insulin now using a genetically modified bacteria that makes the insulin, and then we extract that, and we have this very pure drug. So that's just an example of how widely usable this technology is. And I think there's also some experiments going on now with trying to help people who have a particular disease called sickle cell anemia and trying to genetically modify their red blood cells so that they don't have that defect and actually doing it in people. It's a very new technique and just being very experimental at this point, but that's the level of technology that we have with this.
So again, what's it like?
It's exciting.
It's exciting.
Exciting. Science is exciting.
Sweet. We have definitely the last one.
Okay,
Do you like your job?
I like my job.
We like our job. Okay, sweet. That's it. Thank you so much,
Bye.
guys.